Hello, welcome to lecture number 33. This one is on still in the area of motivation. We're going to be studying sexual behavior and sexual differentiation, and this lecture is on hormones and sexual differentiation. So this is a lecture that will serve as a basis for understanding issues like sexual orientation, homosexuality, both male and female, and give us some context for trying to understand the extent to which our underlying genetic predispositions, as well as the impact of hormones and the environment on our behavior, on our sexual motivation, our sexual behavior. So it's a key topic in the area of behavioral neuroscience, and I hope to be able to provide you with some clarity in terms of a number of the very controversial issues that surround the study of the biology of sexual behavior. So again, we begin our assessment by acknowledging the fact that there's been, a great deal of work that has been done in this area on lower animals, far less that has been done in the case of human beings. And we want to try, of course, to apply what we know with lower animals to the case of humans. Sometimes we can do that, sometimes we can't do that. But it is an area of research that I think has some surprises and indeed is very important for various social policy decisions that have to do with gay rights, for example, and same-sex marriage. So these are important issues and ones that we will get into as we go along. I like to start out by emphasizing what I call a very important take-home message. And that take-home message is this. And again, if you understand what I'm going to tell you now, you'll have an easy time understanding a lot of the research that we're going to be talking about. So our brain and our behavior is basically female. During early life, there are certain biological events that occur that are going to direct sexual differentiation either in a male fashion or a female fashion. But basically, our brain and our behavior starts out as female. And what happens during early life is that when the hormone testosterone, which is the primary male hormone, if that is being secreted during early life, then it's really going to push everything into a male direction, into a masculine direction. So again, our brain and our behavior starts out basically as female. And when testosterone gets added during development, meaning the presence of the Y chromosome is directing this, then you're going to get male-like behavior, masculine behavior. A second important take-home message is this: our reproductive organs and our external morphology are bipotential in the same way in which our brain is bipotential. Our reproductive organs are bipotential. During early life, when if the genetic code is XY, meaning the male genetic code, this means testosterone is going to be added and your reproductive organs are going to develop in a male-like fashion. If there's no testosterone, which is in the case of the female, the XX condition, then what you're going to get is a development of female reproductive organs and external morphology. So both the brain and our reproductive organs essentially are bipotential. So if you have these two concepts down, then I think this is, important for your understanding of this whole area of sexual differentiation, our understanding of later on of sexual orientation. I think that this will be very helpful to constantly be thinking about these two take-home messages that I've elucidated to you on this particular slide. Okay, let's talk a little bit now in terms of the development of the ovary and the testes and what happens again during early life. And at 6 weeks after conception, the gonad of the XX, female, XY, male, they're identical. What you see here, this inner part that you see here of this bipotential structure, this is called the medulla, and the outer part that you see here is referred to as the cortex. Okay. Now, if the genetic code is XX, meaning female, what is going to start to happen is this. Again, there's no Y chromosome present. So the cortex of that primordial gonad, you see right here, that is going to begin to develop, and it's going to develop into the female reproductive organ that we call the ovary. If the genetic code is XY, on the other hand, what you instead are going to see is that instead of this cortex developing, instead you're going to have the medulla developing. That's under the influence of the Y chromosome. So the medulla of the primordial gonad develops into the testes. So again, can't emphasize enough, this is coming from a single structure. And that single structure, again, that we call the primordial gonad, is going to develop in this manner that we see here when the genetic code is XX, and it's going to develop in this manner that we see here, that is as a testes, when the genetic code is XY. But again, they start out the same. Okay. Now, let's take a look at the internal reproductive organs. When we take a look at 6 weeks of life, you know, all human fetuses have the antecedents, meaning the beginnings of both the male, which we call the Wolffian system. Here's the Wolffian system that we see right here in the blue. Okay. And the female system, the Mullerian system that we see here. Okay, in the yellowish orange. Okay. So again, what we have now are the antecedents of both male and female reproductive ducts. If the genetic code is XY, then what is going to happen is that Wolffian system is going to develop further. And again, what you're going to have now is secretion of testosterone from the testes, and also a substance that's called Mullerian inhibiting substance that's going to cause this Mullerian system that we see right here, it's going to cause it to degenerate and go away. Okay. If, on the other hand, the genetic code is XX, you're going to see a development of the Mullerian system. Here is the uterus. Okay, here's the uterine tubes, the fallopian tubes. Here is the ovary that we see here. Here's the upper part of the vagina. And again, the ovaries are here. So it's the absence of testosterone then that leads to the development of the Mullerian system into the female reproductive ducts. And the Wolffian system fails to develop. Okay, it becomes vestigial. So again, in the case of the male, we're getting development of the genetic code of XY, testosterone being secreted. We have the development of the vas deferens that we see here on each side. We have the development of the testes that we see here, and the scrotum that we see here. Okay, so again, this is what things look like in terms of the internal reproductive organs. Now, external reproductive organs, again, 6 weeks of conception. What is happening is that you have the antecedents of both male and female external anatomy. This is the glands that you see here. This is the urethral fold that you see here. These are lateral bodies. And this is the labioscrotal swelling that you see right here. Okay, so if the genetic code is XY, you're going to have the development of this bipotential structure into that of a male-like structure, which in this case, you see the beginning of the development of the penis. And here's the head of the penis, here's the shaft of the penis, here's the scrotum that you see right here, and the anus. So this again is under the XY condition where testosterone is being secreted. In the case of the female, on the other hand, what you observe is that from this structure, this bipotential structure, you're going to see the development of female external anatomy. Here's the clitoral hood right here. Here's the clitoris. Here's the labia minora. And here's the labia majora that you see here. And here's the anus. So again, testosterone, the genetic code is programming an awful lot of this, determining an awful lot of this. And again, this is happening during very early life. So if we take a look at the two primary theories of sexual differentiation, one is saying that it's nature, that's the driving factor. It's these hormones that are acting on the brain and on peripheral tissue that's causing development in terms of physical characteristics as well as psychological characteristics and gender identity and gender role and sexual preference. On the other hand, the other major theory in this area is saying that it's nurture. Nurture is the driving factor. Parents assign a child as male or female, they raise them accordingly, and that's determining, what their sexual orientation is going to be, what their sexual preferences are going to be later in life. So again, we come back to that controversy that we see all along in the field of psychology and behavioral neurosciences. How much is nature, how much is nurture. So there's a theory that has driven an awful lot of the research in this area, and it's simply called the organization activation theory of testosterone effects upon behavior. It postulates that there's two periods of time, one during very early life, prenatal and early life, in which testosterone is exerting an effect. It's having an organizing effect on the brain. It's organizing the brain in a masculine direction. And then we have another period of time during pubertal and adult life where testosterone is activating various behaviors, masculine behaviors. So again, this is called the organization activation theory of testosterone effects upon behavior. And essentially what it's saying is testosterone is having dual effects. When it is present, it's masculinizing the brain, okay, it's promoting male characteristics, behavioral and physical, but it's doing a second thing as well. It's defeminizing the brain. It's suppressing female characteristics. So when we take a look at this and we combine it with this other part of development that we call the activational phase, testosterone is exerting its effect during early life, but it's also exerting its effect during pubertal and adult life in order to activate these behaviors that have been organized at a very early period during brain development. So again, this theory has really driven an awful lot of the research that has been done in this area. Now, let's talk a little bit about some research that was done back in the late 1960s. By a psychologist at Emory University by the name of Dave Edwards, who was very interested in exploring sex differences in behavior. And he chose as his model for sex differences in behavior the study of male rats and mice who are very aggressive in comparison to females who are not very aggressive at all, at least when those females are in a virgin state where they're not carrying fetuses and are not in any kind of a reproductive state. So this is a very well-known sex difference that's seen in many mammals where males are much more aggressive than our females. And this is something that he wanted to pursue in terms of trying to understand. And indeed, he postulated that this was probably due, at least in part, to the fact that males develop with this hormone testosterone and females generally don't. Some really important studies, and I want to talk about two of them. Essentially, what he's doing in these studies is he's changing the early hormone environment. In the case of males, he's taking their testes away during very early life. These are mice that he's working with. And in the case of females, what he's doing very early in life is giving them a single injection of the hormone testosterone. So let's see what happens in these two experiments. Again, in the first experiment, he's taking male mice, castrating them very early in life, that is taking away their testes, their source of testosterone. And then once they get on up into adult life, he tests them for aggression after he starts giving them testosterone during this adult period of time. And what he finds is that these male mice that had been deprived of testosterone at a very early age, these male mice, no matter how much testosterone they were given, they would rarely, if ever, exhibit any aggressive behavior. So that was the first experiment. In the second experiment, what Edwards did was to take female mice on the day of birth and give them a single injection of the hormone testosterone, the masculinizing hormone testosterone, and then allow them to grow up into adult life, start giving them more testosterone, and examine them for their aggressive behavior. And what he finds is they behave just like normal males, and they exhibited extremely high levels of aggression. So this is one of the first indications then that, sex differences in behavior could be altered by simply changing the early hormone environment. And Edwards was one of the first ones to really document this with a sexually differentiated behavior like aggression. So now take a look at this chart that you see here, and let's follow up a little bit with some of the other interesting experiments that were done. And let's look at the left-hand portion of this slide first. If we took a rat, let's say, and this is a male rat, and we simply allowed that male rat to grow up into adult life and then examine it for its behavior, we would see that that male would exhibit a normal reproductive behavior in terms of sexual behavior. Here you see a male that is mounting a female and is exhibiting normal male sexual behaviors. But what if you took that male and instead of allowing it to grow up normally, you castrated that male, took away its testes, and then allowed it to grow up into adult life, and then started giving that male the female hormone estrogen? Well, what would happen is this. Those genetic males, again, who did not have the benefit of the exposure to testosterone during early life, once they were given estrogen, these males would exhibit female-like behavior. What you see here is a female exhibiting what is called lordosis. That's the intense arching of the back that you see in females when they are experiencing high levels of the hormone estrogen. So you can make a male into a female by castrating it during early life and then giving it the hormone estrogen during adult life. Let's now take a look at what happens with females. In a normal female, if we allowed that female simply to grow up into adult life, that female would exhibit normal levels of sexual behavior, would exhibit this lordosis response that we see right here. But if we took that female and instead gave that female just a single injection of testosterone during early life, right, that female would grow up to be male-like in its behavior. And as once we start giving it testosterone during adult life, that activational period, it would instead show this mounting behavior that is very typical of what we see in the case of a male. So Testosterone, what's it doing? It's masculinizing and it's defeminizing, okay, the brain. So this is a summary really of a lot of research that's been done in this area, the area of sexual differentiation. But there's so much more to this picture, and I want to make sure that everybody, you know, understands this before we start getting into some of the human data and the human literature. This slide is a very interesting one. It shows the work, the classic work that was done in this area by a researcher at UCLA by the name of Roger Gorski. On the left-hand portion of the slide, what you see is a brain slice. A brain slice in which you can easily see this particular area that we have highlighted right here, which is near the third ventricle. It is showing a nucleus which is called the sexually dimorphic nucleus. If we take a look at what a male ordinarily looks like, that sexually dimorphic nucleus is very large. If you take a look at it in the case of the female, it's quite small. And this is a well-known sex difference in brain structure. And one of the things that we know is that testosterone is very important for this sexually dimorphic nucleus that we see right here. And the fact that males have this hormone during early life, excuse me, and females don't, apparently you're getting development of this brain area in very different directions. So take a look now at this portion of the slide. What we're going to do here is we're going to give female rats a single injection of testosterone early in life, and we're going to take a look at what their brain looks like during adult life. And indeed, one of the things that we find is that the brain of these genetic females, once they're given testosterone during early life, it causes the growth of that sexually dimorphic nucleus such that their brain looks very much like the brain of a male. And again, here's a control animal that you see right here that was not injected with the testosterone, it was injected with a placebo instead. Take a look now at male rats. What happens when we castrate a male rat during early life? Well, what it does is it reduces the size of that sexually dimorphic nucleus. Compare this to the two control groups that you see right here. What this is indicating is that this area of the brain, this so-called sexually dimorphic nucleus, is responsive to the hormone testosterone. That's why this brain area is enlarged. That's why it gets bigger. And indeed, we know so much more now about this area of the brain and the contribution that it makes to sexually dimorphic behavior. Another piece of research, very important in this area, was work that was done some years ago over in Britain, in London, by Raisman and Field, in which not only are they looking at sex differences in the brain, they're looking at it microscopically in terms of how synapses connect with one another, how neurons connect with one another. And if you take a look at the sexually dimorphic nucleus, again, under an electron microscope, you can see single cells. And if you take a look at that area of the brain of a female, what you see are what we call axodendritic synapses, where axons are actually terminating on dendrites. This is an axodendritic synapse that you see right here, something that's very characteristic of females. Again, when we examine the sexually dimorphic nucleus. Males, on the other hand, have mostly axosomatic synapses, where axons are terminating instead on the cell body, on the soma. Now, that's very interesting. And Raisman and Field asked the question, well, what would happen if we change the early hormone environment? where we took a genetic female and exposed that genetic female to testosterone, or if we took a genetic male and took away the testes during early life, would the brain of that female now, of that male, that genetic male, now look more like the brain of a female? Interesting. So that's what they did. They took males, castrated them during early life, and now what we found is that they show mainly this axodendritic synapsing pattern, again, which is more characteristic of females. So again, castrate the males, got no testosterone. Again, we do this during early life. We get this female pattern of synapsing that you see here. If you do the reverse, take a female that's been injected with testosterone during early life, it will show the male pattern, the axosomatic pattern. So you can change how these connections are being made in the brain, either axodendritic or axosomatic, based upon the early hormone environment. You can change that early hormone environment by giving a female testosterone during early life, or taking a male's testosterone away by castrating it. And again, you get very pronounced changes in terms of these synapses as a consequence of this. So let's talk a little bit now about the activation of sexual behavior by steroid hormones. And again, a little bit of review here. In the case of the female, we're talking about the ovaries. Here's the ovaries that you see here, right? Fallopian tube and the uterus, the cervix, and the vagina. And the two primary hormones that are being secreted are estrogen and progesterone. In the male, on the other hand, the primary hormone secreting glands are the testes. They're secreting testosterone and dihydrotestosterone. Collectively, they are called androgens. So again, when you take a look at the anatomy of the male and the female in terms of the two primary endocrine secreting glands, the ovaries and the testes. Here's the testes in the male that you see right here. This sex difference that we have in terms of hormonal secretions, we want to know a little bit more about the role that they may play in terms of activating sexually differentiated behavior. So a little bit about hormones and these hormones being secreted by the ovaries and the testes. These are what are called steroid hormones, and they have very pervasive effects upon our physiology and our behavior. Once those hormones are secreted and they get into the blood, they reach cells in many different parts of the body. Again, they're diffusing through membranes in order to get to target areas. When they find a target area, when they find a cell, they move inside the cell into the nucleus. They bind with very specific receptors in the nucleus. And then a process occurs that's called transcription, in which we're essentially producing new proteins as a consequence of that. These steroid hormones act on the brain, they act upon peripheral tissue, they act on just about every single organ in the body. Their effects are pervasive. When we take a look at hormones and male sexual behavior, and again, we're going to take a comparative look at this, a comparative examination of this. Here's a male that has approached a female. This female is exhibiting the lordosis behavior, which helps the male, facilitates penetration on the part of the male, and the male mounts the female, intromits with the female, and then ultimately ejaculates. And this is the whole sequence of events that we see here in the male is very much dependent upon the presence of the hormone testosterone. So sexual activity in the male is what we call acyclic. Once a male starts to go through puberty, you see the onset of sexual activity. And once that occurs, a male is more or less ready to engage in sexual behavior all the time. Females, as we will see, show a different pattern. They are cyclic. They show changes in terms of their arousal and changes in terms of their willingness and their readiness to want to exhibit sexual activity. So what we know then from a vast amount of research in many different mammals is that when a male is castrated, let's say castrated just prior to the time of puberty, they show no interest in exhibiting sexual behavior with a female. If they are given what we call replacement therapy with testosterone, where they're actually injected with testosterone, even though they've been castrated, that testosterone is enough to promote normal levels of sexual behavior in that male. What we also know is that the brain area of the hypothalamus, remember we talked about that sexually dimorphic nucleus, implants in this part of the brain, implants of testosterone in the hypothalamus, will activate sexual behavior in a male that has been castrated. So it doesn't have any testosterone, but you can cause that male to exhibit normal male-like sexual behavior simply by implanting very small amounts of testosterone into the hypothalamus. What we also know is that the presence of testosterone in early life is really essential. So you have this dual role of testosterone during early life, setting in motion this organizational process, and then when that hormone is present during adult life, it's activating sexual behavior. So it's very much the case that in male sexual behavior in lower animals, which is acyclic, that dual action, that is the organization of the brain during early life, activation of the brain during later life, all of this being done by testosterone is absolutely crucial. Let me do a little bit of a departure here by talking about an interesting technique that more and more is being used to explore the effects of various hormones on a variety of different behaviors, not just sexual behavior. This is something that's called delivery pumps or osmotic pumps. They are designed to deliver small amounts, physiological amounts of hormones, as well as drugs, if we choose to use drugs over an extended amount of time. And here you see one of these pumps right here, very small. They can be implanted under the skin. And what they do is they constantly release very small amounts of testosterone that mimic what you would ordinarily see being secreted by a male. So again, they are used for a variety of different purposes. We're talking about their usage in experiments that are designed to look at testosterone and its relationship to sexually differentiated behaviors, but they're used for a variety of different purposes in terms of delivery of drugs for perhaps in the treatment of an individual that's schizophrenic or an individual that's hypertensive. So again, I just wanted to make you aware of this particular technique. It is one that is used more and more in the field of behavioral neuroscience in order to deliver various chemicals, whether it's hormones or some kind of drug. So let's talk a little bit now about the female and what we call the estrous cycle. This is in lower animals. In the case of the human being, we call it the menstrual cycle. On the left-hand portion of the slide, you see the hormonal changes that take place over the course of a typical 28-day cycle in the human female. And one of the things that I want to call your attention to is what happens in terms of this hormone right here, the one that's in the blue. This is what is called estradiol that is being secreted by the ovaries. You see an increase that's occurring during what we call the follicular phase right here, which comes as a precursor to ovulation when an egg is actually being produced in the female. Typically, this is what you see in humans, and it's followed a short time later by increases in progesterone that you see here. So again, this is something that is happening just really at the time of ovulation in terms of the secretion of progesterone. If you take a look at some of the important behaviors that you see in a laboratory animal, laboratory rat, you can measure this response that's called lordosis that you see right here. This is an animal, a female, that's not exhibiting any lordosis at all. If you just simply stimulate the flank area of the female, one of the things that you observe is that when a female is sexually active, sexually ready, they exhibit this intense arching of their back in response to stimulation of the flank area. Well, we know now that this is a response that's very much related to hormones. And indeed, this dramatic change that takes place in estrogen followed by progesterone. So look at research that's been done in this area and do a little contrasting and comparing in terms of what we know about the female in comparison to the male. Sexual behavior in the female is cyclic. In the case of a laboratory rat, they're going through their cycle about every 5 days. In the case of the human being, it's every 28 days. In the case of the female rat, it's about 4 to 5 days is their complete cycle. And it's only during a relatively short period of time, maybe about 12 to 24-hour period of time, during that cycle, when they're ovulating, that they are sexually active and will exhibit that lordosis response and will allow males to engage in sexual activity with them. So one of the things that we know is that if we were to take a female rat and take away her ovaries, a process that we call ovariectomy, that's going to eliminate the sexual behavior of that female. If we give them back their hormones by way of estrogen and progesterone, maybe we use some of those osmotic pumps that I talked about, that female will show perfectly normal levels of sexual activity. What we also know is that the hypothalamus is a coordinating point for those two hormones, estrogen and progesterone, that when we put those hormones into the brain, into a female that no longer has her ovaries, that's going to cause that female to show perfectly normal levels of sexual behavior. What we also know is that if we take away testosterone from a genetic male, that genetic male will respond to estrogen and progesterone during adult life by showing that lordosis reflex. Give a genetic female testosterone during early life, and then give that female estrogen and progesterone during adult life, she will not respond to that hormone by exhibiting lordosis. So again, what you see is this real strict dependence upon the presence or absence of testosterone, determining a great deal of what's going to happen in terms of a male being a male and a female being a female. So here's a very interesting animal model. This is simply called the estrogen receptor knockout mouse. You will see this acronym here, ERKO, ERKO, the ERKO mouse. Again, this is what's called the knockout mouse. We've genetically modified these animals such that the genes that ordinarily are involved in the development of the part of the brain that has estrogen receptors, this has been knocked out. Okay, it's no longer there. And one of the things that we know is that those Urco mice are very, very different in terms of their behavior because they don't have that estrogen receptor. Take a look at this work that was done by a Japanese researcher by the name of Sonoko Ogawa. She has been working with these female Urco mice. Here's a female Urco mouse that you see right here. They have no estrogen receptors and they exhibit no sexual behavior. Okay, can't get them to show that lordosis behavior, even if you give them huge amounts of estrogen. They're simply incapable of responding to that hormone. So it's been a very useful model for trying to understand the underlying biology of sexual activity in the female. So let's just talk about one other slide here. We're going to start getting into some of the human work in our next lecture. But this shows the circuitry that's involved in female sexual behavior and male sexual behavior. And take a look at this. Let's first take a look at female sexual behavior circuitry. That's in the green that you see right here. This is the ventromedial nucleus of the hypothalamus. And the circuitry for female sexual behavior extends back here to what we call the periaqueductal gray area. Okay? In the case of male sexual behavior, which we see in this yellowish, orangish color scheme, the circuitry extends from the medial preoptic area that we see here back to the lateral tegmental field. Okay, but notice this area right here where there is some overlap. Okay. This is very interesting, and indeed, when we get to, some of the interesting work that's been done on sexual orientation in humans, and understand that the brains of males and females, it's not an all or nothing kind of, binary relationship, but it may be that males and females do share some of the same circuitry, okay, that's involved in sexual behavior. So this is where hormones are acting to influence behavior, to influence sexual behavior in both males and in females. And we will, explore this a little bit more in our upcoming lecture.